So thank you very much for the invitation for me to come here and to share with you some of our knowledge on this terrible Ceratocystis wilt that we recently described in kiwifruit in Brazil. So I'm very pleased that I could come, and as I told some of you, coming to New Zealand was one of my dreams. So thank you very much for that, okay? Sonia asked me to deliver some very basic and very practical knowledge that on this disease. So I tried to put that way, and I will try to speak slowly and clearly. That's the content of my presentation. So let's see. So that's Brazil and New Zealand. So we are quite far, but we have many problems and also solutions of mutual interest. Okay. That's Brazil. That's the state where I live in, and that's my city in the southeast of Brazil, about 4 km from Rio de Janeiro, that most of you know, especially because of the Carnival, all right? Most maybe have heard about Carnival in Brazil. And the Federal University is in Vicosa, and I was full professor until recently. And in February this year, I retired, but I continue as a volunteer professor, okay? And I'm planning to keep going on this research for a few more years. So that's the university campus, an overview of the university. It's a very small university. We have about 17,000 students only, so it's a pretty small university, but it's one of the most, one of the best, the three top universities in agriculture in Brazil, probably the first. Ceratocystis fimbriata was first described in the United States, in New Jersey, in 1890 on causing black rot on sweet potato. You call here as kumara, all right? Okay? So in Brazil, the fungus was described in 1935 on Crotalaria juncea. It was described as Ceratostomella, and 3 years later, this disease, a disease on mango called mal de Recife, was described and reported as Diplodia recifense, but 12 years later, this famous mycologist changed the name for Ceratocystis fimbriata. So that's how the initial reports of the fungus of this disease in Brazil. Ceratocystis fimbriata is a soil-borne pathogen, and it produces different types of structures, but the main structure is the perithecia containing at the apex a mass of ascospores. Okay, ascospores, and also produce endoconidia and also aleuroconidia. So it has 3 types of spores, okay? Ascospores, endoconidia, and aleuroconidia. Aleuroconidia are a type of resistant form of the pathogen to survive under adverse conditions of the environment. And the fungus can grow very easily in standard culture medium. Maybe a question that you may know, how to identify Ceratocystis wilt. First, by macroscopic observations of aleuroconidia in the xylem vessels. Second, baiting using sandwich in carrot slices, by morphological characteristics of the fungus, and also DNA sequence analysis. So these all four are the main way to identify the fungus. Microscopic observation of Alleroconidia in the xylem vessels, you can see here. Okay? But the problem is, not always we can find this structure. This structure forms especially in more resistant plant material. So there is a limitation to identify the fungus using this technique. In general, the most used method is baiting and isolating the fungus from infected host tissues using sandwich and carrot slice. So we take a piece of infected host tissue, we put between two carrot slices, and we leave for about 10 days, and then the fungus can come up, and you can see here the mass of ascospores at the top of the perithecia, and then we can isolate the fungus easily. So that's the most useful method. The problem is very low success, maybe only 30% of success to identify the fungus, to get the fungus, because we may have bacterial rot from the carrots. Another problem is low competitive capacity of the fungus, so the fungus does not survive very long. And also, because in some cases, the carrots may have some fungicides, and then we believe that these fungicides may limit the fungal growth. So currently, we are trying to use organic carrots to, okay? As you know, the increasing of international trade and the international transit of people has facilitated the invasion of insects and pathogens. Okay? You can see here the commercial airways and the world map. Okay? So, sanitary inspection at airports and ports must be improved and keep going. It's very important. Here you have some papers of my colleagues from South Africa showing the risk that we may have by introducing new pathogens in some disease-free areas. How does the fungus can be dispersed? So, especially this fungus is a soil-borne pathogen, and it can be dispersed mainly by packing materials like pallets, okay, logs, okay, wood chips, souvenirs, okay? So the fungus can be dispersed on this, and even doing treatment for this this material, sometimes it does not reach the fungus inside these packing materials. It can also be spread by root pieces, okay, like colocasia or sweet potato, and as well as by cuttings and mini cuttings, and also by knives, pruners, saws, and so on. But human beings are the only long-distance vectors of C. Fimbriata, okay? So it's not like rust, because rust, the spores can move easily by the wind. By Ceratocystis fimbriata, no, it's much more difficult to spread. The fungus can also, in Brazil, we have proved that it can be introduced in a new area by cuttings collected in the field from infected plant material to make cuttings. So we collect the shoots and we prepare the cuttings, and in the nursery the cuttings can root, but we can introduce in asymptomatic cuttings to the field. either from macro cuttings that we collect in the field or also from mini cuttings in the nursery, because that's the new technique in Brazil. Instead of collecting the shoots from the field, now we collect in the special hedges in the nursery, okay? But in both cases, we can disperse the fungus to the field. The fungus can also be spread by bark and Ambrosia beetle. Bark beetle, it's very effective in some plants like mango, cacao, but we don't have this problem in eucalyptus, not even in kiwi. We don't have much insect activity, okay? In the bark beetle, the Ambrosia beetle can spread the fungus, especially by frass. The frass that comes from the galleries can spread the fungus to different areas. Ceratocystis fimbriata has a very wide geographical distribution. C. fimbriata from sweet potato or from kumara, okay? It's distributed in different parts of the world, but it is just a unique genotype. Just one genotype is spread, and it's also present here in New Zealand, but it's highly specific. It only affects sweet potato, okay? Ceratocystis fimbriata also on eucalyptus has been found in different places, either in South America, Africa, and also here in Southeast Asia, okay? And also on mango, okay, in Brazil, Oman, and Pakistan. On acacia, Brazil, Indonesia, and South Africa, and also on pomegranates, especially here in India and south of China. So this is important to understand this map, because here is New Zealand. So we don't have the distance is not so great. It's great, but it's not so much. Okay, so there is a risk to be introduced especially from this place. Okay. Ceratocystis fimbriata has a very wide host range. It infects different species like mango, kiwi, but it does not infect the fruits. Okay, the fruit is healthy. Okay, but infects the plant. Colocasia, edible fig, rubber tree. Okay, eucalyptus and teak. I had one of the two main crops in Brazil. The most important symptoms of the disease, the radial darkening of the xylem. It's a very important symptom in the wood material. And also, here you see the tree death. How to control, Ceratocystis wilt in eucalyptus. So here you can see how bad the disease can be. And nature teaches us how to control the disease. This is a plantation, a 6-year plantation. Our rotation age in Brazil varies from 5 to 7 years. And this is about 6-year old plantation. And here you have a resistant clone. And here you have a susceptible clone. So why don't I plant this clone instead of this one? Okay? So during my career, I developed a method to screen for resistance in young plant material. And we have here our company, graduated company from the business incubator company of the university. So we have facilities, infrastructure to do screening for resistance, okay? And Simon has visited us recently, 2 weeks ago, 3 weeks ago, and he could see our facilities in the company. So what we do, we inoculate the fungus, and we leave it for to incubate for about 30 to 40 days, and the susceptible plant material has to be discarded, and the resistant one can be used. So in 2004, we published this paper showing the variability of eucalyptus, different clones. Here you have different clones, and here you have the lesion size on inoculated cuttings. Then we have a lot of variability. Now, we have material highly resistant or resistant, and some clones highly susceptible or susceptible. So this clone should not be planted. We can plant these ones, okay? So we published this paper on plant disease, okay? We have screened several clones, and more than 1,000 clones that we have screened, 55% are resistant and 45% susceptible. So it's possible to select for resistance for eucalyptus. Please keep in mind this number, because on kiwi fruit, it's much different. Okay? Before screening for resistance, we have to understand the variability in aggressiveness of the pathogen. We have to select the correct isolate to do the screening. In here you have different clones, eucalyptus clones, inoculated with different isolates. So let's see for this clone, if I use each one of these isolates, I would say that this clone is resistant because we have very little disease. But this isolate here, it infects this clone. The same here. For this clone, these, and these isolates do not do anything, but others do. So it's very important to select the best isolate. In here, for example, this clone was resistant to all isolates tested, as well as these clones. So these clones we use as for checks, to compare, okay, in our inoculation tests. We also use, for the inoculation, we also use a very highly susceptible clone like this one, okay? So before doing the screening, it's very important to understand the variability in the fungal population in terms of aggressiveness, and to select the best isolate to do the screening. Ceratocystis in tick, following what we learned on eucalyptus, we also are doing on tick. So we do the inoculation. In here we have a resistant clone, and here we have a susceptible clone. Okay? So we have also variability in tick for resistant. On mango, what happened on mango? This is part of the work that we have done for Oman, sultanate of Oman. This disease was introduced there a few years ago, and then we had a contract to screen plant material for resistance. So we inoculated the same way, okay? And out of 300 cultivars, of mango, only 34 were resistant to the disease. So it's not so easy as for eucalyptus or teak. Here is Ceratocystis on Acacia manjo. I have been in Indonesia 2 or 3 times, and this disease was recently introduced to New Zealand, to Indonesia. Sorry. And in Acacia manjo, maybe not a viable species in Southeast Asia because of the inexistence of resistant genotypes to an introduced strain of Ceratocystis fimbriata. There is no resistant clones of Acacia manjo. So, strains of the fungus from Oman, Pakistan, India, and Indonesia strains would be very a threat to you here, okay? Especially from mango, pomegranate, Dalbergia sisal, and acacia, okay? And in acacia, by luck, there are some other species which are resistant to this disease, okay? But not manjo. Manjo is highly susceptible. But how about the disease on kiwi, on kiwi fruit, okay? So we have a very small kiwi plantation in Brazil. And the kiwi plantations are in Rio Grande do Sul, okay, state, and Santa Catarina, in these 2 states only, okay? And here is, you can see here the disease in Santa Catarina, for example, this picture from there. And they don't plant kiwi anymore in Santa Catarina because of this disease. Okay. So we published this disease a few years ago, and apparently this disease was in Brazil much longer than that, and they confused this disease probably with Phytophthora, because most consultants that on kiwi were from Chile, where they have Phytophthora. But then we found out that it was not Phytophthora, it was Ceratocystis fimbriata. So the fungus is a soil-borne pathogen, and most infections arise from the soil, okay? And it affects the host tissue and cause wilting. Why do they infect plants wilt and die? Maybe toxins production by the fungus, but we don't know yet, probably, but also because the interruption of the xylem sac, okay? So that's the main symptoms of the disease. I have shown this already. Black spots underneath the bark, okay? And because the xylem is interrupted, the translocation of the xylem, and then you can have wilted fruits like this, but it does not infect the fruit, okay? It only affects the stem, all right? And you can see some patches of dead plants, okay? So you see patches of circles of dead plants in the orchard. Elaine has seen this once in Brazil, all right? That's the circle of dead plants, all right? So, in average, 25 to 30 plants die every year because of the disease. So in 4 years, the orchard is all gone. Okay? And the growers, the kiwi growers, are replacing the crop by other crops like chayote. How you know this plant material? It's chayote. Okay? Corn or grape. So they are replacing because of replacing the crop, the kiwi crop because of the disease. Where do the fungus come from in Brazil? So it's a native there. So it comes from the Cerrado type of vegetation, Atlantic forest, and Amazon rainforest. So we have seen this disease from the north to the south until Uruguay. So it's spread all over the country. What are the main sources of inoculum to kiwi fruit? So naturally infested soil through the roots, asymptomatic infected plant material, grafting, and pruning tools. We have shown the dissemination of the fungus among orchards, and we using microsatellite markers, we proved that from this nursery, this genotype was found in different nurseries, except in this one, because this nursery used to sell the cuttings, or the grafted plants. Okay? So it's, that's how it can move in the orchards by infected plant material. So we have published also this paper. And this to show you the relationships of the fungus from kiwi with other host plants, mango, eucalyptus, and rubber tree. Okay? So it's very important to understand if the fungus from other host species infect kiwi. So that's a question that we have to prove in the near future. So how to control the disease in kiwi? So first of all, is pathogen exclusion. That's what we are doing here, to avoid the introduction of the pathogen. That's the main thing. But if the fungus comes in, so we have to use some culture practices. Wound avoidance, wound dressings, latex paint to pruning wounds, removal of symptomatic plant and stumps, stem injection of systemic fungicides. I just put here, but I don't believe on that. I don't believe that fungicide will control the systemic disease. We believe on planting resistant genotypes or resistant rootstock. For me, that's the main point. So we have assessed the resistance of some kiwi. So we have inoculated them. There's a resistant plant material, that's a searchable one. We also measure the lesion in the xylem. So first of all, for eucalyptus, we also have to understand the variability of the fungus in the kiwi plants, okay, the population of the fungus of kiwi. So we have tested different isolates of the fungus, and here you can see different isolates. So these 5 isolates were the most aggressive ones than others. So look at the importance of select the correct isolate. If I use this isolate, maybe I don't have the proper results, okay? So it's very important to understand the variability in the fungal population. So what we did, to minimize the isolate cultivar interaction, because there is interaction of the isolate and cultivar, we did a mixture of inoculum, inoculate a mixture of isolates. And then you can see here that the mixture of isolates was similar to one of the most aggressive isolates, okay? So currently, we are doing a mixture, a bulk of inoculum. So doing a book of inoculum, we inoculate some kiwi cultivars as part of this project here, okay? And you can see here that all cultivars were susceptible, but Chieftain and Bruno were the most resistant cultivars, okay, which is good. So looking at the cultivars that we have, most of them that we have tested are susceptible, okay? But what to do if all tested cultivars are susceptible? So we, using the experience that we have on eucalyptus, when we inoculate different clones or using seedlings, progenies, we have variability. So my hypothesis was, would I have resistant plant material tested from progenies, from seedlings? Okay? So we inoculate a couple of seedlings of Bruno, and then you can see here that some are resistant. And Simon could see this a few weeks ago, that research that we are going on. Actually, we are doing this part of the project, Sonia, ahead, okay? Okay? So, problem is, the number of resistant genotypes is quite low. It's very hard. It's very hard, okay? It's not like eucalyptus. It's not like teak. It's much harder, okay? But I think if I would have a wild kiwi from China, maybe, I think we can improve this, this number, okay? So it's very, very hard to get. So we had 7 plants out of over 600, okay? And out of this, only 4 left. We got only 4 now because 3 others died by other things. So these 4 clones are sent to Rio Grande do Sul to be tested in the field, because it's very important also to see if the resistance will keep on in the field, okay? So I made a contract with a grower there, Silverstream, which also has a partner of Zespri, okay? So these are the resistant Bruno clones, okay? And these are the screen that we are still trying to increase this number. So in conclusion, Ceratocystis fimbriata wilt, it's a very serious lethal disease in kiwi fruit as well as in other crops. In some orchards, 20 to 30% of the plants may die every year. So in 4 years, the orchard is all gone, okay? Infested soil is the primary inoculum source, but the fungus can also be spread by cultural practices like pruning tools and so on. All kiwi cultivars so far tested are susceptible. Open pollinated seedlings of Bruno segregate for resistance, but the frequency of resistant plants is very low, but it's possible. Resistant Bruno clones may be used as rootstock, but other cultural practices must be implemented to obtain disease-free plants. Okay, so it's very important also to other practices. So that's my team, my current team in forest pathology. Okay. Oh, someone else here. Okay. And that's some of my students and also people that work for me. And most of the people that I have in the lab, the technicians and so on, are paid by the forest companies. Okay, so I don't have public workers, okay, it's from the government. So they are paid by the private industry. So that's my collaborators, okay? Tom Harrington and others, and Mark Templeton. We started a collaboration a few years ago, and also other colleagues, okay? And that's a current partnership with Zespri to develop this research, and Dr. Mark Templeton from Plant Food and Research. So thank you very much for that, okay? And this visit, recent visit to kiwi Ceratocystis infected orchards in 2015. That's Elaine, that's Mike, all right? And Thomas Harrington. That's another view. I just a visit in Vicosa, in my company. And also we have had visitors from Lincoln University every year. In November now, the 14th, another group of students and professors will be going to the company and to the university, and also to visit our company. And that's the most recent visit of Simon and Kathy, okay? And that's my son, Rafael, okay? Okay, so thank you very much, okay? How about the time? Was okay?
Yep, very good.
Okay.
All right. Thank you very much, Acelino. As you saw up there, AgMart have played a major part in sponsoring Acelino's visit to us. So just a huge thank you for AgMart for making his presence here today possible. So we've got about 10 minutes for questions, so just remember, please wait for the microphone before you ask. So have we got any questions for Acelino?
Thank you for a lovely talk, Acelino. I was really interested in the disease cycle. So I understand that it's mainly soil-borne, but obviously they produce ascospores, and you were talking about wound protection. So I wondered if you could comment on the relative impact of foliar or ascospore infection.
I think that's a good point. Actually, the most effects come from the soil. And because this fungus, it does not reproduce on the plant material in general, except if you cut when you harvest, then you can have ascospores formed on the infected plant material. And then it can be dispersed by wind and especially with rain. But in general, the most effects come from the soil, okay? Okay? You understand? Okay? Sure. Yes.
I'm a plant breeder, so I'm interested in your suggestion of getting wild resistant material from China. You know, that seems to me the path that's going to yield the best results if you can go down there.
Mm-hmm.
Any comment on that?
Mm-hmm. Yeah, because of the variability of the commercial varieties of the cultivars is very low. So I believe that if I select from a wild plant material, may have a higher frequency of resistant plants. It has happened in several other diseases like tomato, and also for some viruses. So I think that's one of the ways to increase the frequency of resistant plant materials to get, to select, to inoculate seedlings of wild plants from, wild plant material from China, I think. That's my hypothesis, but I'm not sure. But at least we see that there is variability. It's very low, but on Bruno, it's very low, but to me, if you get wild plant material, we may have a higher frequency. I hope. As a breeder, you know that wild plant material may be a better source of resistance. All right?
That's what I found.
Yes, yes.
It's more of a comment rather than a question. For those who don't know me, my name is Miguel. I've been managing kiwifruit orchards in Waikato for about 12 years. Born and raised in Brazil, where they grow kiwifruit, and that small area where he was pointing before. My family doesn't grow kiwifruit, we grow citrus, but I've been to that area. I know what is his name? The importer. The kiwifruit importer that you're talking to. Silverstream, yeah. I know
Silverstream.
I know Silverstream well.
Silverstream, yes.
And I actually met some growers. In relation to your questions, that they because they know that in New Zealand we grow a lot of kiwifruit and we know all about kiwifruit, and they said, oh, what can we do to stop? Looking at their orchards, it's quite difficult to sort of give them a recommendation on what to do, but I would, there were places where there were isolated plants infected, and they don't sterilize the tools.
No.
They're not used to. It's like when PSA arrived in New Zealand. You know, it's like it was a mental shift about, oh, it's important to clean the tools and so on. And also, imagine if a soil-borne disease and you're going up and down with the tractors and trailers, and so you kind of spreading that to various different ways. I guess my comment is I've been there and I saw the disaster that it caused it. Like I said, a lot of growers aren't going to a different crop because it's not a main growing for kiwifruit. They just end up planting something else. Yeah, and I'm really happy to see you, Acelino. For one of the first times I've seen a Brazilian scientist coming over and speaking about kiwifruit. Yeah, so thank you.
Okay, thanks, Miguel. But I think it's important to try also some fungicides to protect the plant. So actually, we have the research plan to also to test that, okay? To see if some fungicides can avoid the fungal infection, okay? Yes?
I was just a little bit interested in, you said that it comes from your native forests, the disease.
Yes.
What sort of impacts is it having in your native forests?
Okay. We have actually a disease on Carapa guianensis in the Amazon rainforest, but it's different a type of disease. It's called dumping off. So the fungus kills the seedlings. So I believe that it may reduce the biodiversity of this native plant, which is Carapa guianensis. It's very nice native Amazon plant, okay, tree, okay, but it causes another disease, okay. It causes dumping off. It does not cause wilting. And I was talking to Samuel. Samuel is doing the annotation of the genome of this, and he said that this is one of the most different isolates. Okay, Samuel? And also we have found the fungus in the Cerrado type. Cerrado is more a draw-out type of vegetation. Okay, it occurs in more dried areas. So it also infects a very nice a tree, which is pequi. Okay, you have heard about pequi, Miguel? Pequi.
I heard about it.
Yes, and so it has the fungus has killed a lot of plants. Okay, on this native Cerrado type of vegetation. Okay, so.
So how do you think that's spreading? Is it root-to-root contact?
I think it's a soil-borne. Okay, to me, the fungus is endemic in these areas. Okay, so in native forest, is because there is an equilibrium of pathogen and the plants. So we don't have an epidemic So you see one plant here, another plant there that is infected, okay? So it may affect, but it's not like a crop, okay? In crop, it's, okay, it's much more the impact. Yes, sure.
Thank you for coming to New Zealand, and
thank
we hope the weather improves.
Thank you very much.
I missed earlier, was it the same genotype on sweet potato, kumara, or different genotype from kiwifruit? Pathogen?
It's different. The genotype of, maybe Samuel can answer better. The genotype of sweet potato is an unique genotype all over the world. Okay, it's just one genotype spread. And Samuel is comparing the genome, right, Samuel? Can you?
We're having a presentation after morning.
Yeah. So far, we are comparing the genome structure and genome content from these isolates, and the isolate from kumara is quite similar to the isolate from sweet potato from United States, but both isolates is quite different from our kiwifruit isolates from Brazil.
These differences are common in the pathogen population, and many people try to describe new species. I am more conserved. To me, it's a variability within the fungal population, the same species. But maybe some mycologists in the future will describe new species, like the people from South Africa, they like a lot to describe new species. But to me, it's the same species, just a variability. Like here, we are, each of us are different, all right? So the same thing occurs in the population.
Professor, thank you very much for your talk.
Thanks.
Question, the isolates which impact through on kiwifruit, have they only been found in Brazil, or are they present in China or any other kiwifruit producing nation?
So far, only in Brazil. But I believe that because the fungus is in China now, according to all the other species, just a question of time, probably. But we have to understand if the fungus from these other crops infect the kiwi. That's one of my proposals is to test that, okay?